Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today we're going to be talking about enzyme kinetics, which includes the michaelis-menten equation, lineweaver burk plots, and more. But first I want to do a quick review of what we talked about last episode, where we talked about the 6 enzyme classifications. I want to give a few more common examples for each one, just to really solidify the learning. So oxidative reductases we talked about, which catalyze oxidation and reduction reactions. This is where electrons are transferred from one molecule to another. These include dehydrogenases, oxidases, and reductases. For example, alcohol dehydrogenase catalyzes the oxidation of alcohols to aldehydes or ketones. Then we have transferases. These catalyze the transfer of a functional group, like methyl or phosphate group, from one molecule to another. An example of this is alkynases or methyltransferases, such as hexokinase. Then we have hydrolases. These catalyze hydrolysis reactions. These include proteases, lipases, esterases, and phosphatases. Then we have lyases. These catalyze the breaking of various chemical bonds by means other than hydrolysis and oxidation. And this often forms a new double bond or new ring structure. Examples of these are decarboxylases and synthases, such as pyruvate decarboxylase, which removes a carboxyl group from pyruvate, releasing carbon dioxide. Then we have isomerases, which catalyze the rearrangement of atoms within a molecule. This changes the molecule from one isomer to another. Example of this is racemases, epimerases, and mutases. For example, phosphoglucomutase, which facilitates the transfer of a phosphate group within a glucose molecule. And finally, we have ligases. This is the enzyme that catalyzes the joining of two molecules, forming a new chemical bond, and often hydrolysis is happening side by side. This hydrolysis of ATP needs to happen, as joining with ligases often requires the input of energy. And examples of these are synthetases and carboxylases. Notable example, like we talked about last time, is DNA ligase. Now let's actually move on to enzyme kinetics. So enzyme kinetics refers to the study of the rates at which enzyme catalyzed reactions occur and how these rates are affected by various factors, especially substrate concentration and enzyme concentration. One of the most important features of enzyme kinetics is how reaction velocity, which is denoted as V, changes as substrate concentration increases. So at low substrate concentrations, there's a linear increase of the rate of the reaction with substrate concentration, and this is because many active sites are available. But as substrate concentration rises, the enzyme molecules become saturated, meaning active sites get occupied. So adding more substrate here does not increase reaction significantly. At this point, the reaction rate approaches its maximum velocity, which is denoted as Vmax. An enzyme's maximum velocity, or Vmax, is the point at which the enzyme is working at full capacity. So let me break this down in an analogy. I live on an island, and to get to this island, you need to take a ferry. And there are always taxis lined up outside the ferry station. So if we think of the ferry passengers as a substrate who want to get home, that's the product, then the taxis would be the enzymes. Now at first, when there are less people than taxis, then the reaction rate would increase linearly with more people. But if there are more people than taxis, then the people would have to wait because the taxis would all be saturated. At this point, it doesn't matter how many more people come. If there's 100, if there's 1,000, the reaction is going as fast as it can, and it's at its maximum velocity, or Vmax, as it is working at full capacity. Now this is described by the michaelis-menten equation. This says that V, which is a reaction velocity, is equal to Vmax multiplied by substrate concentration divided by Km plus substrate concentration. Now Km is the Michaelis constant. This is the substrate concentration at which reaction rate is half of Vmax. So essentially, Km is the substrate concentration at which half of the enzyme's active sites are full. So the concentration of people when half the taxis are full. Now, Km is used to compare enzymes. It provides insight into the enzyme's affinity for its substrate. A higher Km indicates a lower affinity because it requires more substrate to be half saturated, whereas a low Km indicates high affinity because the enzyme reaches half of Vmax at a low substrate concentration. The Km is an intrinsic property of the enzyme substrate system and cannot be altered by changing the concentration of substrate or enzyme. So in our example, if we assume the bus is a different enzyme, we can compare them. If more people tend to get on the bus, then it has a higher affinity, which means that it will get half saturated with a lower concentration of people, giving it a lower Km, making it more efficient. Whereas for the taxi to be half saturated, there needs to be a much higher concentration of people, giving it a higher Km, making it less efficient. Now the michaelis-menten relationship generally graphs as a hyperbola. So when substrate concentration is less than Km, changes in substrate concentration will greatly affect the reaction rate. But at high substrate concentrations exceeding Km, the reaction rate increases much slower as it approaches Vmax. At Vmax, it becomes independent of substrate concentration. On the y-axis of this graph is reaction velocity, which is V as given by the equation, and the x-axis is substrate concentration. So to find Km from here, first you want to know where your Vmax is. Then you want to know where your half Vmax is, and that will be on the y-axis, okay? And then you will have a line, a slope or something. You draw a line out to the point that intercepts with the y-intercept, where it's at half Vmax, and then you draw straight down. And the point at the x-axis, that's half Vmax, will be your Km. And this is because your x-axis is substrate concentration. Now this hyperbola and everything can get kind of confusing. So to better analyze this kinetic data, the lineweaver burk plot can be used, or the double reciprocal plot. The double reciprocal plot is exactly as it sounds. It takes the reciprocal, or 1 over, of the x and y axis. So the michaelis-menten, the x-axis is substrate concentration, and the y-axis is V, or reaction rate. And so in a lineweaver burk plot, the x-axis is 1 over substrate concentration, and the y-axis is 1 over V, or 1 over reaction rate. And once you plot this data on this, you will get a linear line, and it will be much easier to interpret. So the x and y-intercept can also tell us data. The x-intercept will be -1 over Km, the y-intercept will be 1 over Vmax, and the slope will be Km over Vmax. Although this data is less intuitive than the hyperbolic michaelis-menten plot, lineweaver burk plots are useful for identifying types of enzyme inhibition because Vmax and Km can be compared without estimation. And we'll talk more about inhibition in the next episode. Just to finish off this episode, we'll talk about cooperativity. This occurs in enzymes with multiple active sites. These will show a sigmoidal curve or an S-shaped curve on the michaelis-menten curve. So subunits and enzymes can exist as either a low affinity tense state, which is called the T state, or a high affinity relaxed state, which is called an R state. Binding of the substrate encourages the transition of other subunits from the T state to R state, and this increases the likelihood of substrate binding to the other subunits. Conversely, the loss of a subunit can promote R state back to T state, and this leads to dissociation. Enzymes showing cooperative kinetics are often regulatory enzymes in pathways like phosphofructokinase and glycolysis. That's it for today. Next time we'll talk about an enzyme inhibition.